Hello, this is BIO461, and today we're going to be talking about the kidney. We're going to walk through how the kidneys filter blood and control what stays and what leaves our body. We'll talk about filtration, reabsorption, how water and solutes move, and we'll also talk a little bit about how hormones like ADH or energy and aldosterone kind of fine tune everything within the kidney. So on the left side of this board, here's our kidney. This little pathway going down here is called the ureter, and then this is the urethra, and this is where we pee. We pee out of that. And so it's connected to the kidney, and these striped lines are called the medulla. This is just kind of the anatomy of the kidney. And right off of those striped lines with these little bubble looking things, we call these nephrons. So these are nephrons, and blood flows toward, I drew that in red, towards the kidney through arterioles, and then away from the kidney through like venous things like veins and stuff like that. And so that's kind of the anatomy of the kidney. This is a filtering machine. So over on the right side of this page, the first thing that happens with the kidney is filtration, and it happens in the renal corpuscle. And the renal corpuscle, or the Bowman's capsule, are these circle things that we see, the round things. The little green things inside of it, we call it the glomerulus, as you can see there. Blood enters the glomerulus through afferent arterioles and leaves the efferent arterioles. So it goes through kind of like this, afferent through efferent. I know it's kind of crammed, but that's how it kind of works. As blood flows through the glomerular capillaries, pressure forces water and small solutes out of the blood and into the Bowman's capsule. Things like the ions like sodium, glucose, amino acids, and urea or salt are small enough to pass through. It's pretty specific. But larger proteins and like blood cells are too big, they stay in the blood. The filtration membrane itself has three layers: the endothelium, the basement membrane, which blocks large proteins, and the podocyte filtration membrane. Podocyte filtration slits, which basically fine tunes what passes through into the kidney and stuff. Filtration happens because of pressure differences in the kidney. The glomerular hydrostatic pressure pushes fluid out, and the capsular hydrostatic pressure and blood colloid osmotic pressure push everything back. The result is a small net filtration pressure, which is about 10 millimeters of mercury that drives the filtration. So not very much pressure, but it is a small space. The next thing I want to talk about a little bit is GFR and regulation. GFR stands for glomerular filtration rate. It's basically how much fluid is filtered each minute, about 125 milliliters of mercury per minute in a healthy person, like a normal person. Let's see if I can find a good thing to remember this. Give me 1 second. Okay, so let's see here. Maybe I could draw this out for us. So this is a complex nephron. We can simplify it in a very easy way. Make it easier to look at. So I'll draw it like this. So here's our nephron, and then we're going to draw the afferent and efferent pathways. So this is efferent over here. I hope you can see it. This is afferent pathways. Kind of drew that quick. But there we go. Oh, and there's a glomerulus. So I drew it in green before. I'll draw it in green again. The glomerulus kind of lines the bottom of this right here. So this is glomerulus. There you are. So that's a simple way to draw a nephron. Blood enters afferent, leaves efferent. there's the glomerulus. So that's one simple way. The kidneys can regulate GFR in two main ways. I just drew it myself. Two main ways. First, through the myogenic mechanism, where the afferent arteriole constricts when pressure is too high. It constricts, so tightens, and that will protect the glomerulus. And the second way that it can regulate GFR is through the tubuloglomerular feedback. And the tubuloglomerular feedback is pretty simple. There are maculodental cells, or little muscles, like right on the top of this thing. And so, let's see. So, tubuloglomerular feedback consists of this. GFR stands for glomerular filtration rate, and so decreased GFR equals maculodental cells which release
if I can have you guys
nitric oxide, and then that eventually increases GFR, and then that leads to negative feedback.
Next we have on the left is the reading center and the study skills center.
And then that'll just keep the cycle going. And so that's a little bit about tubuloglomerular feedback. Hopefully you can hear me okay. We can also measure how well the kidneys basically filter using something called clearance, and that basically means clearance is how quickly a substance is removed from the plasma in our blood. It uses substances like inulin or creatine, where we can estimate the GFR a little bit. Okay, so that's a little bit about GFR and how that works. Next, we are going to talk about reabsorption. I'm going to erase this. And we're going to personally talk about reabsorption for the kidney in one of its tubules. So it has lots of tubules. It has like the collecting tubule and a few others. But one of the biggest ones is the proximal convoluted tubule, or PCT. I'm going to draw this out. We're going to talk about a lot of different ones. So this is the way I learned it. So one rule about tubules, there's always water moving through the tubules. And there's a lot of ions that also move through the tubules through channels. So water always moves through, and it goes into the blood. And so the blood is like right here. Let's say our blood is there. And let's say all of our ions are here. And so we're going to have like water, sodium, calcium, potassium, bicarbonate, lactate, amino acids, glucose, and chlorine. And wherever water or wherever sodium goes, water follows. And so there's a lot of different channels. I'll kind of simplify each one, but there's a channel that purposefully moves only glucose and sodium in. So sodium goes in and glucose goes into this channel. So that's one of the channels, and those things get pushed back. The main thing about all three of these ions, they eventually, lactate, amino acids, and glucose all get pulled into the blood after. That's very specific. And so, lactate, amino acids, and glucose. Okay. Now the next channel puts sodium and lactate through. We'll draw that red. So sodium also goes through this channel. Lactate goes through this one as well. And then the final one, sodium and amino acids go through. Water follows. Sodium. Back, or sodium and amino acids, not lactate. Okay. And all the rest of the ions just kind of move through. There's a glucose channel down here that moves glucose out a little bit quicker. And then water moves through consistently. So this is H2O. I drew that kind of sloppy. But this is the proximal convoluted tubule, moves a lot of ions through. Sodium is the key player. Sodium is actively pumped out of the tubule cells into the blood using sodium-potassium pumps. We talked about that early on. That creates a gradient, so other solutes can follow. Glucose and amino acids hitch a ride with sodium as well, using these transporters, and we call that secondary active transport. As sodium and other solutes leave the tubule, water follows osmosis. We call that obligatory water reabsorption, I believe. And then chloride and potassium also follow along, so chloride and potassium. Where was I? Chloride and potassium follow along passively because of electrical and concentration gradients. Now these transporters can eventually get overwhelmed because there's a lot of information, as you can see here. Sodium always goes through, lactate, amino acids, and glucose follow through, and eventually get into the blood. And so there's something called renal threshold that comes in when these channels are overwhelmed. For example, if blood glucose gets too high, like in diabetes, for example, the transporters for glucose become saturated. When that happens, not all the glucose can be reabsorbed, so it starts to appear in urine and stuff like that. And so that isn't necessarily that good. We're going to talk a little bit about the loop of Henle now. We did talk about that before. I drew it like down here on one of the kidneys. And so after the PCT, which stands for, let me remind you, the proximal convoluted tubule, after the PCT, the loop of Henle, or the filtration moves into the loop of Henle, or the loop of Henle picks up filtration. I'm going to erase this. Actually, no, I'll leave this up here for a second. This is where the kidney creates concentration gradients. It's super important for controlling like water balance and stuff like that. That's what the loop of Henle is all about. The descending limb is permeable to water, but not salt. So water moves out and into the salty medulla, which is that area that I drew on the kidney with all the little mini stripes. And the filtrate becomes more concentrated. The ascending limb of the nephron, I might draw this out so that you can see it again. So do you remember seeing something like this with like a circle and then it like goes down and up and then like down, up. There's a circle here. So these are our nephrons. Loop of Henle is down here. And ascending is up here. And this is ascending limb. And descending limb is like down here. Like this area. Descending.
The last thing I want to mention is this. So again.
So that's kind of what it looked like a little bit. That's super simple, but there you go. And so the ascending limb is the opposite side. It pumps out sodium, it pumps out potassium and chloride, but water can't follow, so the filtrate becomes more diluted. This back and forth setup between ascending limb and descending limb is called the countercurrent multiplier, and it's what builds and maintains a salty environment of the medulla. So if you take like a bite out of the kidney, don't do that. But if you did, it would taste very salty. It's super salty. Water tries to follow, but it can't. Okay, next we're going to talk about another tubule of the kidney. So the way I drew my tubules was this, remember?
Just remember that this is the resource that is provided to you by tithing funds. This is the resource that the
And
school
so we're going to
pays
draw the
or
distal
has
and the
for
collecting
you.
tubule. So this is distal and collecting tubule.
There's a lot of people
Um,
that go to other
and
universities and they
It
have to pay
basically does the fine
$80
tuning
per
of everything.
100.
Sodium is reabsorbed and potassium is secreted. So like sodium potassium pumps up the wazoo. And especially under the influence of hormones. So hormones can definitely affect this. And so it uses ADH, of course. There's sodium that goes in. So there's sodium that's in, and then it secretes our potassium. So it secretes our potassium, like we can see. And then water does not move through, actually. So we're going to pretend like there's a big barrier here. Water tries. So there's water that comes by. This is another reason why it's so salty. Water comes by, it is deflected, just like that. Yes. Oh, I forgot one thing. So here's the blood, and there's a hormone called aldosterone that comes from the blood and goes into the collecting tubule. So we call this aldosterone. And we'll be talking about that more. There's aldosterone that comes in, and so does ADH. There's an ADH receptor here that collects it.
Thank you guys again so much for listening. I hope you guys have a great rest of your day.
ADH. So it picks up some hormones on the way that affect it, and there is also sodium over in his blood here. Sodium, there's a bunch here. Water does not move through, so it's very ionic and salty and stuff like that. Okay, so that's a little bit about the collecting and distal tubules. Next, we are going to talk about renin and aldosterone and ADH to build off of it. And so when blood pressure or sodium levels drop, there's something called juxtamedullary cells, and they release renin. Renin covers angiotensin 2, or angiotensinogen and angiotensin 2. So I'm going to draw out this little pathway. And so, let's see here.
You could ask
I can
Ina
draw
too.
this.
If Ina approves it, then you're good.
Okay, so give me 1 second while I draw this out. We're going to draw a kidney simply. This is a kidney. Blood flows through. It releases renin. And then renin goes to the liver. And the liver, so let's say the liver is right here. We're going to draw that in blue. So that's kind of what the liver looks like. Renin goes to the liver. The liver spits out something called angiotensinogen. Holy cow, I'm sorry. Angiotensinogen. And then angiotensinogen is transformed or converted into angiotensin 1 or Ang1, and then Ang1 is then converted into angiotensin 2 or Ang2. I guess we can also use red for that, it's similar. Angiotensin 2 has a lot of purpose. It causes constriction, which raises the blood pressure. It triggers the release of aldosterone from the adrenal cortex. Adrenal cortex is this area. Adrenal gland is like an afro on top of the kidney that kind of looks like that. And so aldosterone acts on basically the main or the principal cells in the distal tubule, so the one that's farther away, and the collecting duct, the one that we just drew earlier, if that makes sense. Maybe I can label this with angiotensin or aldosterone. Maybe I can show you that aldosterone comes from this. Hormones. It increases the number of sodium potassium pumps and sodium channels. So more sodium gets reabsorbed back into the blood. Potassium is secreted and water follows the sodium because wherever sodium goes, water goes. This raises the blood pressure and blood volume in your blood. Meanwhile, when the body needs to conserve water, the posterior pituitary, which is a gland, it releases ADH. So does angiotensin. Increases ADH, which is energy, and it just releases ADH, so it gives you energy. ADH acts on collecting duct cells, adding aquaporin channels, which move water through to the membranes. This allows water to move through, to move out of the filtrate and back into the bloodstream, basically concentrating the urine so that you're peeing more water than like blood or salt. Because if you're peeing more salt or sugar, then that could lead to kidney stone. And so finally, I want to talk a little bit about, one more thing about aldosterone. It increases ADH, increases your thirst, so you want to drink more water. This marker is being so bad. Increases thirst. And it also increases constriction. Or vasoconstriction. Okay. I got that all out. There you go. Renin out of the kidney, liver, turns it into angiotensin, nudgin. Ang1, Ang2 increases aldosterone, which is a hormone, increases ADH, which is also a hormone, increases thirst. You want to drink water. Blood flows through, vasoconstricts the blood, or water flows through, vasoconstricts the blood, goes out your ureter so that you can pee it out, and you have more water in there than anything else. So finally, we're going to talk a little bit about the urine, concentrating or diluting it. The medullary concentration gradient that the loop of Henle, remember down here, creates is what makes ADH effective. If ADH levels are high, then water leaves the collecting duct and the urine becomes concentrated. If ADH levels are low, water stays in the filtrate and the urine is diluted from water. So together, the loop of Henle sets up the gradient and ADH decides how much water is reclaimed. To summarize, finally, with the end of the kidneys, filtration happens at the glomerulus, that thing that we were drawing out earlier, and filtration happens at the glomerulus. Reabsorption happens mainly at the proximal tubule. The loop of Henle builds the gradient, and hormones like ADH and aldosterone fine-tune the water and the salt balance so that you can pee water, basically, mostly water. Altogether, the kidneys constantly adjust what we keep and what we lose to maintain homeostasis so that our bodies can live and be okay. Kidneys are very powerful, but they are also very complex. So that is what we learned over the week. Next week, we're going to be talking about respiratory, so lungs and breathing and stuff, and I'm very excited for that. We'll see you then.